So in this video, we're going to look at what I call the specific immune system or the specific immune response. In another video I have here on YouTube, I talk about what's called the non-specific immune response. These are things that your body does every day. And they're non-specific because they don't know that this is a bad thing, a pathogen, a virus, a bacteria. They just know it doesn't have your MHC protein or your special marker protein on it. So they destroy it. So things like your skin. Your skin has acidic residue on it. Your skin has salts on it. That's going to kill things that land on it. One part of your skin doesn't kill just bacteria. One part of your skin doesn't just kill viruses. So your skin is part of what we call your non-specific immune response. Mucus in your body is what we call the non-specific immune response. And so this goes in a lot more detail in another video that I have here on my YouTube channel about that. But let's say the non-specific immune response doesn't work. And when this happens, your specific immune system has to kick in. Now, your non-specific immune responses, your surface membrane barriers, mucus, things called killer cells, they are going to work 24 hours a day, 7 days a week. They do not turn off. However, your specific immune system has to be turned on. This does not run all the time because luckily we don't need it all the time. We're not constantly facing bad guys. We don't have infections. Now, you have pathogens in you and on you all the time. So technically you are infected all the time. But just because you're infected doesn't mean you're sick, because 90% of the time your non-specific immune response will get rid of whatever it is that might make you sick. So in the case of the specific immune response, this is when, let's say, you've had a cut or you have an infection, and this has been in your body for several days, and your body has been trying to get rid of it, and nothing has happened yet. So basically, your non-specific immune response, even though it's working as hard as it cannot get rid of this bad guy. It can't stop the infection. So this is about 5 or 6 days after you've already started to, or excuse me, after you've been infected. So at this point in time, you're like, okay, I don't feel good. And it's very possible you have a fever. And what's happening with specific immune response is it's having to turn on. And it's having to figure out, okay, what is it that's attacking me? Okay. Something has been inside of me. It's causing a release of histamine. I have cells that are being damaged. You know, what is causing this to happen? Because your body has to fight different pathogens differently. If you have a virus, your body's going to fight it one way. If you have a bacteria, your body's going to fight it the other way. So the specific immune response has to be turned on. And this starts when a macrophage does what's called a display. Macrophages are just a common category name we give a bunch of different types of white blood cells. And these white blood cells will destroy your cells that aren't supposed to be there. And so if this little white blood cell realizes, okay, I keep destroying the same thing and it doesn't seem to be getting away, or it doesn't seem to be going away, it will kind of like sacrifice itself, and it will allow that bad guy to infect it, or when it eats the bad guy, it engulfs the bad guy, it takes the bad guy's proteins and coats its surface with it. Remember, every cell in your body has proteins on the surface. And those proteins are called antigens. And that's how your body knows what kind of cell it is. So for example, your liver cells will have one kind of protein on them, and your brain cells have a different kind of protein on them. And that's how your body knows what these cells are. Now, I always draw, when I'm in class, proteins as shapes, because it's very easy for you to distinguish shapes. So let's say that, liver cells have these round shaped proteins on them, and brain cells have square shaped proteins on them. Well, everything in nature, even viruses, have proteins on the outside. And the way that viruses and bacteria look to infect you is they want to put their pieces, their proteins with our proteins like a puzzle. They have to find the correct fit. And so when this macrophage realizes, okay, I keep fighting off this bad guy and it doesn't seem to be going away, it will basically display its marker protein. So it's like you waving a red flag saying, hey, I need help over here. And then we have special cells called helper T cells. Now, when you need help, you dial 911. And when you dial 911, you talk to a dispatcher. And the dispatcher gathers all of your information, and the dispatcher calls the police department, and the dispatcher tells the fire department where to go. Well, your body has dispatchers as well. And these are these things called helper T cells. When cells are damaged, they give off histamine. And we always have cells being damaged in our bodies, so we always have a small level of histamine. But your body realizes when histamine levels start to spike, that's like a 911 call. So when you need help, you dial 911. When your cells need help, they secrete a hormone you've all heard of called histamine. Well, helper T cells are the dispatchers. They recognize this spike in histamine. They recognize that these macrophages are waving these red flags and that they need help. So your helper T cells then will call out the alert and start your specific immune response. So your helper T cells kind of turn on the specific immune response. And at this point in time, your body has to figure out, okay, what is it that I'm fighting off? So just like you dial 911 and you talk to the dispatcher, and then the dispatcher calls the police department and the dispatcher calls the fire department, the helper T cells are going to get this whole thing going. And this is one of the reasons why HIV is so bad. HIV, human immunodeficiency virus, attacks helper T cells. So people who are HIV positive, it's like having a really big city like Raleigh, but only having 5 or 6 dispatchers. So people who are HIV positive, their body has a hard time getting the specific immune response to turn on. It would be like you dialing 911 and the phone rings and rings and rings, and eventually somebody will answer it. Well, when it gets to the point where you don't have enough helper T cells to just do normal everyday activities, at that point in time, this person's considered to have AIDS. And it would be like you dialing 911 and nobody ever answering the phone. But histamine levels have spiked. The dispatcher, the helper T cell, answers the phone, takes down the information, and it's going to start what we call a specific immune response. Now, in the specific immune response, there's 2 parts to it. Just like when you call the dispatcher will then call the police department and the fire department. And the police department's job is to go after the bad guy. The fire department's job is to put out the fire. Well, you have 2 parts of your immune system that act the same way. So in the case of your specific immune response, you have what we call the cell-mediated immune response. This is what I call the fire department. This is made up of T cells, and these cells have to kill our own body cells that have already been infected. Because if our cells are infected, they're damaged, they're not working like they're supposed to, and so we need to destroy them so that this infection will not be spread. So if there's an arsonist on the loose and you call the dispatcher, the fire department is going to come to your house to put out the fire. And so that's what the cell-mediated immune system does, what I call your fire department. So in the case of the cell-mediated immune response, like I said, we have these helper cells, and helper cells are also called T4 cells. And these are your dispatchers. They recognize that these pathogens are in large numbers. And so a lot of times we have a spike in histamine, but again, histamine is a 911 call. And the helper T cells then start the cell-mediated immune response, and it will start something called the humoral immune response. The cell-mediated immune response is calling the fire department to come put out the fire. The humoral response will be calling the police department to come arrest the bad guys. And helper T cells start both of these. So histamine levels have spiked, the dispatcher is taking down the information, and the fire department is on its way to put out the fire. And that is your cell-mediated immune. And so our firemen are what we call killer T cells. So just like when a house is on fire, the firemen will stand back and spray water onto the fire. A lot of your killer T cells act the same way. They don't really come in contact with the bad cells because they don't want to risk being infected themselves. But the killer T cells will recognize infected human cells and cancer cells. Cancer cells are cancerous, meaning that they won't stop dividing, and they tend to have protein mutations that show up on the surface of their cells. And killer T cells can recognize that these proteins aren't the way that they're supposed to be. So just like a fireman will spray water onto the house, killer T cells tend to work by secreting hormones, chemicals. And these chemicals are called perforin. When you perforate something, you punch holes in it. And so the killer T cells themselves won't touch the infected cells. They're going to shoot this hormone at it, this chemical at it, and it eats holes in the cells, which then causes water to rush into the cells and the cells explode. And this is how our body destroys cells that have already been infected. Now, something kind of stands back to make sure that the job is getting done. So we also have what are called suppressor cells or T8 cells. And when you suppress something, you stop it from working. Basically, killer T cells will not only destroy cells that are infected, they'll also start to destroy cells that look like they are good cells. So basically, the killer T cells will also kill cells in the surrounding area, just in case they're infected, but they haven't started to show that yet. And the suppressor T cell's job is to tell these cells, okay, you guys have destroyed enough, the fire's out, there's no more smoke, you can stop doing your job. So in cell-mediated response, the firemen go to the cells that are infected and destroy those. So it's just like the firemen coming to a house and putting the fire out. We also have what we call the humoral response, or the antibody-mediated response. And these are done by a group of cells called B cells. And I call these your police department. And police officers carry weapons, and hopefully they don't have to use those weapons, but those weapons are there to help protect the community and help stop the bad guys. Well, the B cells, or the humoral immunity, are your police department. It's their job, while the fire department is putting out the fire, it's their job to actually find the arsonist. It's their job to capture the arsonist. And they do so with something you've all heard of before, that something called antibodies. And antibodies are going to be like the weapons that these special types of cells called B cells carry. Now, in the case of B cells, like I said, are your police officers. And every police officer carries a weapon, and every weapon is different. So even though you look at a whole bunch of guns, you guys know that there are ballistics. Even though all the guns look the same, when they shoot the bullet, it leaves different markings on the bullet case. So basically, every police officer carries a different gun. You can think of it that way. Well, B cells carry weapons that are called antibodies. And antibodies are actually proteins, and they're always drawn as a Y. One antibody is actually made up of 4 different proteins. And think of these proteins as having shapes. So some antibodies might be square, some antibodies might be circles, some antibodies might be heart-shaped. And these are arrows that we shoot at the bad guy. Now, it's not going to kill the bad guy, but it's going to block the bad guy. Because remember, the bad guys, the pathogens, the viruses and the bacteria are looking to connect to our cells. So it's very much like having a piece of a puzzle. So our B cells are going to shoot these antibodies at the pathogen in order to try to block the pathogen. So if the pathogen has a circle-shaped protein on it, we're going to shoot an arrow that has a C on it so that the C can clasp onto the circle, and it blocks the pathogen. Now the pathogen can't attach to another cell. So we're going to shoot, that says shot, sorry. We're going to shoot antibodies at the pathogens in hopes that it sticks, and then the pathogen is going to be blocked so it can't stick to something else. And like I said, with antibodies are always drawn as a Y, and antibodies themselves are actually made up of different proteins. And so in this picture here, you can see the Y shape. The proteins at the top that form the Y are often called the variable chain. Variable means it changes. And the straight leg portion of the Y is called the constant chain because it doesn't change from antibody to antibody. So we can look at how the chemical makeup of antibodies are. We look at their constant chains. What makes every antibody different is the Y portion. Think of the Y portion, the V, the split portion, the fork in the road, as being a different shape. That's what makes every antibody different. But when you look at the straight line, that's what we call the constant part of the protein. That doesn't vary that much in nature. And what we find is we have 5 different categories of antibodies. The first one is what we call IgM. Ig stands for immunoglobulin. That lets you know that this protein is found in our blood. And I always think of M as being multiple. This guy down here that looks like a star, that is an IgM antibody. It's like having 5 different antibodies in one. So we can assume, or we can pretend, that this antibody right here is a circle, this antibody is a V. Let's see here. This one might be a square, okay, but you can get the idea with shapes. And this one here might be, an upside down triangle. Okay. But by having different variable or different shapes, it's basically like having a Frisbee with 5 different shapes on it. So instead of having 5 different police officers go to an area, we only have to send out 1. So it's like having 5 different antibodies at one time. And so I think of IgM as being multiple. Now we also have IgA. IgA is this double one here. It's like having 2 antibodies at one time. And again, the shape on each antibody would be different. So I'm trying to find a color that actually works. So this end might be, have a square shape to it. And this end might have, this is supposed to be a circle shape to it. And so that's like having 2 different antibodies at one time. IgAs, I think of them as acids. These are secreted. So we find these antibodies in our tears, in our mucus, inside of our mouth, on the surface of our skin. We also have what we call IgG. I think of G as being general. This is the most common type of antibody that we have. And this is the type that you borrow. And what that means is, as a fetus, your mother destroyed pretty much everything for you. Your immune system didn't really have to do a lot. But when you were born, you borrowed some of your mom's antibodies. And this is there so that you have antibodies to use until your immune system can kick in and start making you antibodies. We also have IgE. I think of IgE as eosinophil. Okay. Eosinophils are a special type of white blood cell that carries these type of antibodies. And these types of white blood cells only show up in our blood in large numbers when we're having an allergic reaction. An allergic reaction is kind of your own body overreacting. Allergic reactions, in one way, your body's doing what it's supposed to do. It recognizes that histamine levels have spiked. So it sends out the immune response, it sends the police officers, it sends the fire department. But in allergic reactions, your body is kind of playing a trick on you. The only time histamine levels should spike is when you have cells that are being damaged. When people have allergic reactions, their bodies are like, oh, this is a weird chemical. But peanut oil cannot hurt your cells. Cat dander cannot hurt your cells. And so you have a group of cells called mast cells that just start to make histamine for no reason. And that trigger of histamine causes the specific immune response. And eosinophils will create these antibodies that will actually turn off our own mast cells. So the only time you should have IgE in our body is when you're having an allergic reaction, because this is your body's way of trying to turn off these mast cells that are secreting histamine when they really shouldn't be secreting histamine. And histamine can cause our bodies to make more mucus. That's why when you have, an allergy, your nose runs. But also we have mucous membranes in our breathing tubes. And so that's why people with severe allergic reactions, they, have to be real careful, because it can actually affect, how they breathe. Their tubes could actually swell shut. Now, if you are a white blood cell, if you are a B cell, you will have one of these. You don't carry all 5. So some of your white blood cells, some of your B cells just carry IgMs. Another group of your B cells will just carry IgAs. The last one, however, is carried by all of your white blood cells. It's what's called IgD. I think of IgD as death or IgD as docked. This is an antibody we should never have to use. This antibody never unattaches. If you are so sick that nothing has worked, your white blood cells will sacrifice themselves. And they'll use IgD, this antibody, this docking antibody, to attach to the bad guy and allow the bad guy to infect them. So, like I said, the way that B cells work, different B cells, when your body realizes that there's a bad guy, all these different B cells, you have different B cells with all these different shapes. They basically line up and take turns. So, if I'm the bad guy, the first person would line up and they would shoot these arrows at me. And the arrow might bounce off. If the arrow bounced off, then we know, okay, this is not the weapon that I need to use. And so your body will work your way through until it finds an antibody that sticks. And so it's like, okay, this is the weapon that I need in order to block the bad guy. So we clone that specific type of B cell and we just shoot the antibodies until the bad guys are completely covered. And when they're completely covered, they can't attach to one of our cells and infect it. And then the macrophages can then come and eat the pathogens.